In this video, we're going to go through viral structure. So this is really for AQA biology only. We're going to label the key components of a virus, and we're going to go through the functions of the key components that all viruses have. So first things first, this came up on an exam paper. I don't know whether you've seen it, but viruses are acellular. What does this mean? So many students did not know what acellular meant, but it basically means that viruses are not made of cells. Okay, that's all it means. They are not cells. Now, there are alternative ways that you could have got this mark when this question came up on the exam. So you could have said things like, they do not have cytoplasm. They do not have a cell membrane. Or you could say they have no organelles. They do not have any organelles, okay? Don't just say they don't have membrane-bound organelles, because neither do prokaryotic cells, and they are cells, okay? So bacterial cells or prokaryotic cells, they are cells. They don't have membrane-bound organelles. So viruses, we can't just say they don't have membrane-bound organelles. We have to say they have no organelles at all. Even cytoplasm, they don't have it. Cell membrane, they don't have it. So they are not cells. They are not made of cells, okay? So just let's learn this definition. They are also not living. Why? So they do not carry out metabolic reactions. Or you could give an example, they do not respire. Think all the way back to Year 7 biology when you learned Mrs. Gren. To be classed as a living thing, you have to be able to respire, and viruses do not respire. They do not excrete, something else that we see in Mrs. Gren. Living things have to be able to excrete. They don't need nutrition, which again is the N in Mrs. Gren. They also cannot replicate or reproduce unless they are inside a host cell, because remember, they hijack host cells and they use the host cell's machinery in order to replicate or reproduce. So they cannot do it on their own. So they are not classed as living things, okay? Let's have a look at the general structure of a virus and we'll just label it. So A is pointing to an attachment protein, which we can also call a glycoprotein, but I tend to always call it an attachment protein. B is pointing to the capsid, which is kind of like a protein coat, okay? Now, inside the capsid, we have the genetic material or the genome. Now, this could be DNA. Some viruses have DNA for their genetic material, or it could be RNA. For example, HIV has RNA as its genetic material. Around the outside, we've got a lipid envelope. It's not a cell membrane, because remember, viruses are not cells. They do not have cell membranes. It's a lipid envelope. Not all viruses have these, though. So I wouldn't name that as something, if it asks you to name a structure that all viruses have, do not go for lipid envelope, because some viruses are just genetic material, capsid, and attachment proteins. The pink dots, you might be wondering what they are. Well, this is actually reverse transcriptase. Again, not all viruses have this, okay? But this virus in particular is HIV. And HIV has RNA as its genetic material. So it also needs the enzyme reverse transcriptase, because what reverse transcriptase does is it converts viral RNA into DNA so it can be integrated into the host cell's DNA using another enzyme called integrase. Again, not all viruses have this. They don't have this enzyme. They don't need it if they've got DNA. But HIV needs this enzyme because its genome is made of RNA, so it has to convert that RNA into DNA before it can be inserted into the host cell's DNA. Okay. Let's have a look then at the 3 key features that all viruses have. So all viruses have genetic material, or a genome, we can say. They all have a capsid, which is made of protein, and they all have attachment proteins on their surface. So remember, they don't all have a lipid envelope, and they do not all need reverse transcriptase. Now, in terms of the functions, the genetic material should be quite easy to give the function for that one, because we say it all the time. The genetic material, or the DNA, or the RNA, or the genome, it codes for proteins because the order of bases determines the order of amino acids in the protein. Obviously, this is a virus, so this DNA or RNA will be coding for viral proteins. The capsid, which is made out of protein and surrounds the genetic material, we can say it protects the genetic material, or it stores or it encloses the genetic material, or the RNA, the DNA, or the genome. Finally, the attachment proteins, which are on the surface, they actually attach to receptors on the host cells. And this is how a virus actually enters a host cell. So it uses those receptors, it uses those attachment proteins to bind to receptors. The receptors will be complementary to the attachment protein. And once the virus attaches, then the genetic information and possibly the reverse transcriptase, if needed, will enter the host cell, and then the genetic material will be inserted into the host cell's DNA. An example of this is with HIV. The attachment proteins on HIV attach to receptors called CD4 receptors, which are on the T helper cells, those lymphocytes in our immune system. If you want to learn more about HIV, so if you are taking AQA for A-level biology, I am going to be posting a video very soon on HIV replication. So make sure you come back to check out that video too.